Ever wondered how cells survive extreme temperatures? Their secret lies in their membranes. Let's explore how membrane lipids adapt to different conditions. Cell membranes can exist in two physical states, gel and sol phases. In the gel phase, lipids are tightly packed and movement is minimal. The membrane becomes rigid, less flexible, and less permeable. In contrast, the sol phase is dynamic. Lipids move more freely, giving the membrane a fluid and flexible nature. This shift between gel and sol phases is influenced by temperature and lipid composition. Starting with saturated fatty acids, these chains contain no double bonds, hence only sigma bonds. Because of this, the hydrocarbon chain remains straight and fully extended, allowing free rotation along carbon-carbon bonds. This extended structure minimizes steric hindrance, making the molecule highly stable. Thanks to their straight shape, saturated fatty acids can pack very tightly together, forming what we call a crystalline array, a highly ordered structure. These strong van der Waals interactions result in rigid gel-like membranes with high melting points. Now compare this with unsaturated fatty acids, which contain cis double bonds, a defining structural feature. These pi bonds cannot rotate, which causes a bend of about 30 degrees in the hydrocarbon chain. This kink disrupts the packing of adjacent fatty acids, making the structure disordered and loosely packed. Because of this irregular arrangement, unsaturated fatty acids experience weaker van der Waals interactions, resulting in membranes that are more fluid and have lower melting points. So here comes a quick summary of the differences. These points have already been discussed. Now let's see how organisms adapt their membrane structure based on temperature. At low temperatures, membranes tend to become too rigid, almost gel-like. This rigidity can hinder the movement of proteins and disrupt cellular functions. To counter this, organisms increase the proportion of unsaturated fatty acids in their membranes. The cis double bonds introduce kinks in the hydrocarbon chains, which prevent the lipids from packing too closely. As a result, the membrane stays fluid and functional even in cold conditions. On the other hand, at high temperatures, membranes may become excessively fluid, which can compromise their structural integrity. To stabilize the membrane, organisms incorporate more saturated fatty acids. Their straight chains pack tightly, forming a more ordered and less permeable membrane. This adjustment ensures that membrane proteins remain in place and essential functions like transport and signaling are maintained. Cholesterol plays a crucial role in eukaryotic membranes as a fluidity buffer. At high temperatures, the phospholipids in the membrane gain kinetic energy and move more freely, making the membrane overly fluid and unstable. Cholesterol fits snugly between the phospholipid tails and restricts their movement, helping to stabilize the membrane and reduce permeability. On the other side, at low temperatures, phospholipids tend to pack tightly, making the membrane rigid. Cholesterol disrupts this tight packing by inserting itself between the fatty acid tails, thus preventing a transition into the gel phase. In this way, cholesterol ensures that the membrane remains functional and semi-fluid across varying temperatures, preserving membrane protein function, transport, and signaling pathways. It's this dual action, stabilizing when too fluid and loosening when too rigid, that makes cholesterol an essential component of eukaryotic cell membranes. So in the end, membrane fluidity is everything. Saturated fatty acids make it rigid, unsaturated ones keep it fluid. Cells adjust this balance depending on temperature, and in eukaryotes, cholesterol fine-tunes it even more. It's all about staying functional, no matter the conditions. Thanks for watching and stay tuned for the next video.